glucose. Now, the position of this anomeric alcohol group is really important when we think about linking together two sugars, because oftentimes we are going to link them through this anomeric carbon. All right, we're not going to do it, but biology does it. So let's look. Here's a couple of glucoses together, and we're going to tie them together into a disaccharide. So again, this carbon number 1 is the anomeric carbon, the one that varies in either in the alpha or the beta form. And what we're going to do here is we're going to do an alpha 1,4 linkage, okay? Which means the 1,4 is pretty self-explanatory. We're going to link these sugars together between the 1 position and the 4 position, right? So the 1 position is the anomeric carbon, which could be alpha or beta. The 4 position is over here, which is not anomeric, which means this alcohol group is always in the same position. And the coupling of these two monosaccharides together into a disaccharide is a condensation reaction. We're going to lose water, and the water that we're losing here is outlined in pink. So here we go. We've got one of the oxygens here as the bridging oxygen that bridges the carbon number 1 and the carbon number 4 together. And this is called an alpha linkage. And the reason why it's called an alpha linkage is because the anomeric carbon on carbon number 1, the alcohol group was in the alpha position. Now, how can you tell it's in the alpha position? Well, a clue that I've learned is that if this bridging oxygen here is on the opposite side of the molecule as the alcohol group on carbon number 6, then that's the alpha position. Okay? If the bridging oxygen is on the same side as the alcohol group on carbon 6, that's the beta position. And that's sort of counterintuitive for me. You may be fine with it. Usually I think of alpha is going to be on the same side, beta on the opposite side, but that's the opposite here. Alpha is when the oxygen is on the opposite side. Beta is when it's on the same side. And so we can describe this disaccharide by saying, well, what are the two sugars? These are both glucoses or glucopyranose for the cyclized version. How are they linked together? Well, they're linked between the carbon number 1 and carbon number 4. So this is a 1,4 linkage. And if one of those carbons is anomeric, then we have to describe in what position the alcohol group was when this coupling occurred. And so this is an alpha 1,4 linkage here. And really, we should have an alpha probably designating this 1,4 linkage. Now, this makes a disaccharide of glucose, and a disaccharide of glucose with an alpha 1,4 linkage is the sugar maltose, which you may have heard of before. It's sort of a milk sugar. You may have had a malted shake, or you've eaten Whoppers, those candies that they give out at Halloween. That sweet but sort of different taste is maltose. I like it for a little bit, and then I get sick of it real quick. I can eat about one Whopper, and then I don't want to eat anymore. Okay, so that's how we make disaccharides, and that's really how we make polysaccharides also. We have to designate which carbons are being coupled together and whether we've got an alpha or beta linkage. And so here's some examples of some disaccharides here, again, that you've heard of these things. Lactose is a disaccharide. Sucrose, table sugar, is a disaccharide. Trehalose, you probably haven't heard of before. So let's focus on lactose and sucrose. And let's look at them. Here's lactose. It is a disaccharide of galactose and glucose. Okay, it is a 1,4 linkage, right? We can see here. And is this going to be an alpha or a beta linkage? Well, here's this anomeric carbon. We look, and this bridge here is shown in this really funky way to show that it's sort of a diagonal bridge here. The oxygen's at the top over here, but at the bottom over on the 4th carbon. And so this would be a beta linkage, because if the oxygen is coming from the top, that's on the same side as the alcohol on the 6th, We said the same side is going to be our beta linkage. So this is a beta 1,4 linkage between galactose and glucose, right? Okay, here's sucrose. We've got fructose over here, which is a 5-membered ring, and glucose over here. So here's again our anomeric carbon right there. And we've got a 1,2 linkage, right, between the first carbon and the second carbon of fructose. And we have to designate really whether we've got the alpha form or the beta form, since this is also an anomeric carbon on fructose. And so we've got the alpha form over on the glucose here, because the oxygen is on the opposite side of our alcohol group on the 6th carbon. And this is a beta linkage for fructose, because the oxygen's on the same side as the 6th carbon alcohol group on the fructose. Okay. And trehalose here, let's just do a little test. So try to figure out what type of linkage we've got. We've got 2 of these glucose molecules, right? They're linked between a 1,1, right, which is a pain because both of these are anomeric carbons. So we're going to have to label this linkage in 2 ways. Here we've got the oxygen on the opposite side of the 6th carbon. So that's an alpha right here. What is it over here? Well, here the bond is going up, so the oxygen's here, which is on the opposite side of the 6th carbon here. So we've got another alpha. Even though it looks like this bridge is going back and forth, this is an alpha-alpha linkage here between 2 glucoses. Okay, so those are disaccharides, and we can continue linking them together to form polysaccharides. And these polysaccharides can come in different forms, right? Unbranched, which means the linkages between all the monosaccharides are the same. For instance, they're all alpha 1,4 linkages. These are homopolysaccharides, which means all the monosaccharides are the same. Maybe this is a alpha 1,4 linkages between a bunch of glucoses. Our homopolysaccharides can be branched. Maybe we have a parent chain here of alpha 1,4 linkages, but maybe we have some alpha 1,6 linkages that form branches in our polymer. We can also have heteropolysaccharides that alternate, or maybe it's just random stuff that are branched all over. In lab, we've talked about how a lysozyme can cleave a NAM-NAG polymer. NAM and NAG are both monosaccharides, and they alternate. And so this is a good example of like a NAM-NAG polymer in the bacterial cell wall that lysozyme can cleave. Okay. So why do we link these things together? And especially why do we link glucose together, since we're going to be talking about glucose metabolism? A lot of times it's for storage, right? When we polymerize glucose, we make this long molecule that we can store as glycogen, or plants as starch. And we store it in our liver, and we can access that more easily. Sometimes it increases solubility. When we polymerize and branch, we can access that energy more easily, and we'll see how there's enzymes called alpha amylases that are present in your saliva and your digestive system that can cleave these polymers and release glucose as we need for metabolism. Okay. So here are starch granules in plants and glycogen granules that are in our liver. And these are just homopolymers of glucose, and they all have alpha 1,4 linkages, right? So here's amylose is just an alpha 1,4 linkage of glucose. We can see we've got our 1,4 linkage, and it's an alpha linkage. Oxygen's on the opposite side here, and our anomeric carbon. Amylopectin is another version of that's present in glycogen, and it is an alpha 1,4 linkage of glucose, but every once in a while there's a branch with an alpha 1,6 linkage, right? Here's the 1 carbon, and instead of attaching to the 4, it's going to attach to the 6. When we look at these, uh, amylopectin and glycogen have a really spiral structure that has branches coming off of it. And this allows the glucose and glycogen to be really accessible for metabolism. There's lots of different ends to this molecule that can be accessed by amylose. And so amylose can chew off glucoses one by one, releasing free glucose into the bloodstream through metabolism. And having more ends due to all this branching allows for many enzymes of amylose to attack this one molecule, spitting out lots of glucoses instead of just having a linear molecule where you have to start at one end and go to the other. Okay, so again, we see this spiral molecule forming in these glucose polymers, and probably as a kid you tried to stain different foods with iodine to see if starch was present in there. Starch is just the alpha 1,4 linkage glucose present in plants. And, iodine sort of binds right in the middle of the helix caused by a glucose polymer, and it changes its absorbance, so you can see it turn black, right? Okay, so there's not only alpha-linked glucose polymers in nature, there are beta-linked homopolymers of glucoses. And these we think of as fibrous molecules. And any fiber that you eat in your diet is really composed of these beta-linked homopolymers of glucose. It's cellulose, right? Cellulose is really insoluble, and amylose, the enzyme, does not recognize these beta-linked homopolymers. And so things like cellulose, we can't digest. Our amylose molecule does not recognize the beta 1,4 linkage. It only recognizes alpha 1,4 linkages. And so when plants make cellulose with the beta 1,4 linkages, it's really hard to release the glucose from these molecules of cellulose. And animals that do it, who have plants as their main food source, really takes a lot of energy in order to release glucose from these cellulose molecules. They have to chew it up to increase the surface area. They have bacteria in their gut that really can cleave the beta 1,4 linkages, but it still takes a lot of effort. If you ever looked at a cow, it's just constantly chewing it up, swallowing it, really infecting it with bacteria so that the bacteria can cleave away some of these beta 1,4 linkages, brings it back up, chews it some more. It's really difficult to get that fiber, the glucose, out of these fibrous cellulose chains. And here's the beta 1,4 linkages, right? We could identify this as a 1,4 linkage, and beta because this bridging oxygen is on the same side of the molecule as the 6th carbon. Okay, so most of the hydrogen bonding that's present in these beta 1,4 linkages is occurring within the molecule, and that stabilizes it, making these really stable, really hard to break fibers of beta 1,4 glucose. And it also makes it insoluble because instead of interacting with the water solvent around, these alcohol groups and all the polar natures interacting with itself, making it insoluble in the polar water. Okay. Now, there's a bunch of important polysaccharides. Starch is made of amylose, amylopectin, glycogen, cellulose. Chitin is the exoskeleton of a lot of insects, and it's made of these beta 1,4 linkages between glucose and this modified glucose, right? That has a acetyl group on and an amine on it. Peptidoglycan we've talked about before, or actually we haven't talked about before. We're going to talk about that in just a minute, but this is part of the bacterial cell wall that has these NAG polymers, and this is a structural polysaccharide that's found in joints in humans a lot of times. Hyaluronic acid, you may have seen it in drugstores, people trying to take hyaluronic acid to help with knee pain. It's a lubricating polysaccharide.